CD19 CAR-T treatment has been applied in refractory and relapsed diffuse B-cell lymphoma recently. Durable complete remission can only be seen in 29% to 37% of patients. Could you highlight the benefits and the challenges for this kind of treatment and explain the potential causes for relapse? Thank you.
Thank you. So the potential causes for relapse, I think there are definitely two that have been considered. One is antigen escape, and the other is CAR T-cell exhaustion. And so the Alexander study is using a bicistronic CAR targeting both CD19 and CD22. And thereby using two target antigens, it is designed with the hope of reducing relapse via antigen escape, so CD19 negative relapse. CAR T exhaustion is possibly due to upregulation of PD-1 on the CAR T cells, and there are in vitro data to show this. And therefore, the Alexander study uses pembrolizumab in the conditioning regime to try and reduce CAR T cell exhaustion. Initially, it was introduced on day plus 14 for safety reasons when most of the CAR T expansion had occurred. And then when the safety data were good, the pembrolizumab was moved to day -1, so that there was still the benefit of introducing it, but earlier in the conditioning regime.
As we will know the study, your team reported the first incorporation of dual targeting CAR-T and anti-PD-1 in diffuse B-cell lymphoma. Could you please introduce why AUTO3 is so unique compared with other CAR-Ts? Could you explain the mechanism of adding pembro and dosing of this drug? Thank you.
So AUTO3 is different to other CD19 CAR-Ts. It's a bicistronic CAR, so it uses one vector to introduce two individual CARs, which are specifically designed to target their own CD antigen, so whether it's CD19 and CD22. Most CARs, CD19 CARs, use FMC as their binder, whereas AUTO3 uses humanized HD37. It also uses OX40 and 41BB co-stimulatory domains, which are designed to improve persistence. So the CAR design is unique in the fact it's bicistronic, and also its humanized factors and its co-stimulatory domains. The introduction of pembrolizumab was designed to try and reduce CAR T-cell exhaustion. And obviously, initially, there has to be safety assessment of this. And that's why in the first design of the trial, it was introduced at day 14 when maximal CAR T expansion had occurred. The toxicity of AUTO3, which has been presented, is very low, with no significant CRS and minimal neurotoxicity. And therefore, pembrolizumab was then introduced to day -1. The effects are seen early and the CAR T expansion is early, so therefore it is reasonable to introduce it early into the conditioning regime because it has that durable effect which allows that efficacy of the construct in combination with PD-1 inhibition.
Could you share us the Alexander study, clinical design, and the main results of this study? During the escalating stage, which is the recommended dosing? Thank you.
So the Alexander study is a phase 1, phase 2 study, starting with a 50 million dose, and then dose escalating through doses of 150 million cells with 450 million cells as the maximum dose. And this is now completed. Flu-cy conditioning is standard, and pembrolizumab was initially introduced for 3 doses from day 14 and then brought to day -1. The safety data was the primary endpoint for the phase 1 part of the study, looking at toxicity in the first 75 days. Most of the serious adverse events were hematological and reversible. And of interest, there was no severe cytokine release syndrome, and most CRS was grade 1. Only 17% of patients required tocilizumab, and there was no prophylaxis required. There was only one neurotoxicity event out of the 23 patients dosed, and this was quite atypical. The patient had some facial and muscle weakness, and of interest, the patient had similar episode 10 years previously without a diagnosis reached. So this may be related to this. There was no neurotoxicity at the 150 and above dose or with pembrolizumab. And therefore, because it is felt to be safe, moving forward, the dose range of the recommended phase 2 dose range is 150 million to 450 million cells with day -1 pembrolizumab. And the efficacy has been really encouraging. So there were complete responses seen with all doses delivered. And for the 23 patients, the complete response rate was 48%. When looking at the patients who received more than 150 million cells, the complete response rate was 56%. And this was where the dose range was decided moving forward. For patients who received more than 150 million cells and day -1 pembrolizumab, the complete response rate was 63%. So although early data with the first 23 patients treated, this is safe and the efficacy is very encouraging, especially when we look at the sustained responses that are achieved in comparison to other CAR-Ts where 25% of patients relapse.
Okay, thank you. And I'm interested in this clinical trial because BC Stronic, you chose viral vector encoding both an anti-CD19 and anti-CD22 CAR-T. So it is very interesting design. And I'm wonder what's the difference between this CAR-T and then with the sequential use anti-CD19 CAR-T and anti-CD22 CAR-T. What's the difference between this design?
So this is a bicistronic CAR in comparison to other tandem CARs, which don't have the independent antigen target. So these are two independent CARs delivered with the single vector on one CAR, rather than a tandem CAR, which other centers are using.
Okay, yeah, I think maybe it's very interesting, and I will also wonder why if you use a retroviral vector. Do you have any introduction to us to about how about other vectors using in the CAR-T clinical trials?
So the retroviral vector has been successful in manufacture of all the CARs. All of the patients had sufficient products made as required for the trial, and because it has been successful and sustainable, this is why we have used a retroviral vector.
Thank you. I have the fourth question. How about the CRS and other side effects of the patients who received Auto3 compared with other CAR-T treatment? How do you balance the clinical benefit with risk? Thank you.
So I think that this is an extremely positive aspect of Auto3 is its safety. We see significant CRS and neurotoxicity with the currently available CD19 CARs. Almost a third of patients have these toxicity, and significant numbers need to go to intensive care because of CRS and neurotoxicity. Auto3, in my experience, has been incredibly safe to deliver with no severe CRS and only this one event, which was quite atypical of neurotoxicity. And again, no neurotoxicity seen with the moving forward recommended phase 2 dose. So I think that as well as the early efficacy data being incredibly encouraging, I think the safety data is also encouraging. And because of the safety data, it's allowing us to move to an outpatient-based expansion cohort, which will be opening soon for recruitment.
Thank you. And as we all know, for the CAR-T clinical trials, the severe CRS and the neurotoxicity is the main toxicity of the CAR-Ts. But in your study, the Auto3 didn't have the severe CRS neurotoxicity. I wonder why. Can you explain to us why there's so less toxicity in this clinical trial?
So it's possible that the low levels of toxicity are because of the low cytokine levels that Auto3 seems to stimulate. And there are quite clear in vivo and in vitro data that Auto3 has lower levels in association with CRS compared to other CD19 CARs. And because of the lower cytokine release, I think that could explain the low toxicity, but yet there is adequate cytokine release to cause efficacy in terms of patient response.
I'm also interested in the design of this clinical trial. You use CAR-T first, then use PD-1 antibody. And do you think the PD-1 antibody treatment is very important in this clinical trial? Thank you.
I think so. The mechanism behind CAR-T exhaustion is obviously recognized, possibly due to PD-L1 expression and also upregulation of PD-1 on the CARs. So trying to inhibit that, but in a safe way with PD-1 blockade, appears to and may be the cause for the excellent efficacy data that we are seeing so far.
You have used the 3 doses of pembrolizumab, and then with regimen A, and but in regimen B, you only use 1 dose of pembrolizumab. And how about your opinion about these doses of pembrolizumab? Do you need the 1 year's pembrolizumab treatment, or just only 1 or 3 doses of pembrolizumab? Thank you.
In terms of the half-life of pembrolizumab and the timing of the response and maximal CAR-T expansion, which is within the first 28 days, it appears reasonable to give just 1 dose on day -1 of the conditioning. And therefore, moving forward, especially in view of the low toxicity and excellent response rates, this is why we are moving forward with the day -1 dose only of pembrolizumab in the dose expansion phase.
I also wonder about how about the auto stem cell transplantation with the CAR-T. Some authors use the CAR-T before the stem cell transplantation. Some doctors suggest stem cell transplantation before CAR-T treatment. And how about your opinion? And how about the Alexander study? Do the patient can receive autologous stem cell transplantation in the clinical trial? Thank you.
Previous auto stem cell transplant was not an exclusion criteria for this trial. This trial isn't a treatment which is planned as a bridge to further transplant, whether that be auto or allo. It's because of the durable and sustained complete responses, we would be planning that this treatment alone was adequate for the patient.
Nowadays, the CAR-T treatment is very interesting and very promising in the treatment of relapse and refractory diffuse B-cell lymphoma. And in the Alexander study of Auto3 suggests maybe the use the 2 kinds of CAR-T, CD19 and CD22 dual targeting CAR-T may be very effective in this treatment. And I don't know if you can use 3 or 4 kinds of CAR-T together, not only just the 2 kinds dual. Thank you.
At the moment, I don't have any experience of giving sort of more than 2 dual antigen targeting. I think that, managing to get using just 1 vector, both CARs available is already successful and has got good results. So I think that we know that some of the patients do relapse because of CD19 escape, but that isn't the case for all relapse in diffuse large B-cell lymphoma. So by using a bicistronic CAR, that may try and affect one of the resistance mechanisms, but also using it in combination with checkpoint inhibitors and other approaches.
Thank you. And I have another question about the new CAR T cell treatment. Some also use the maybe CD19 combined with anti-PD-1 CAR T. How about your opinion about the PD-1 CAR T cell lymphoma cell treatment, or you prefer to use the anti-PD-1 antibody? Thank you.
Okay. So in the Alexander study, we're using an anti-PD-1 antibody, so pembrolizumab, and that is because that can block both the PDL ligand 1 and ligand 2 axes, whereas if just using a PDL1, then that doesn't affect the PDL2 axis. So in my opinion, it appears to be using an anti-PD-1 may have better efficacy in terms of trying to reduce CAR T cell exhaustion.
Nowadays, in the lymphoma treatment, there are some dual antibody, such as anti-CD20 antibody combined with anti-CD3 antibody, the dual antibody. How about your opinion about this dual antibody treatment? Thank you.
So looking at bispecific antibody treatments, I think is also very encouraging. And I think that there are some benefits with this in terms of they can be off the shelf treatment, so to speak, rather than having to allow the manufacturing time when the patient can progress. I think, both of these directed therapy, both CAR T cell and bispecific antibodies, look really encouraging for the future. And what would be helpful is if we're able to select the patients who could benefit from each approach whilst minimizing toxicity.
For the CAR T treatment, the CAR T T cell therapy, in fact, was approved in relapsed and refractory diffuse large B-cell lymphoma. But as we all know, for the relapsed and refractory mantle cell lymphoma, also CAR T treatment is very effective, in which is published in the New England Journal of Medicine in last April. But also for the follicular lymphoma, also CAR T is very effective. Do you think maybe CAR T cell lymphoma, CAR T treatment maybe will be the standard of many kinds of refractory relapsed subtypes of lymphoma, not only diffuse large B-cell lymphoma? Thank you.
I think that's difficult. In the UK, at the moment, we are only allowed to use the 2 licensed products for relapsed refractory diffuse large B-cell lymphoma after 2 prior lines of therapy. As yet, we cannot use for low-grade or mantle cell. I think that it obviously has implications on health economics and cost of these expensive therapies, and whether they can be delivered and for how many patients. I am hopeful that they will be an option for many of our patients who have relapsed subtypes of lymphoma, and whether it's a cellular therapy approach or a bispecific anti approach, I think we need more data and longer follow-up for us to really know that answer.
And for the CAR T treatment, there is a problem about the duration of the response. Maybe some patient will relapse after CAR T treatment. And how about your opinion? How to prolong the duration of the response of the CAR T cell lymphoma? Thank you. CAR T treatment in lymphoma.
So durability of response is really what we're trying to improve upon now with the current CAR T and the current CAR T designs. It appears to be possibly related to the co-stimulatory domain that is chosen. It may be because of CAR T exhaustion. Other concerns are heavily pre-treated patients. You may be able to make less efficacious CARs from them because they've had so much heavy pre-treatment with many lines of therapy. So I think that as we further gain experience with using the different CARs available and make sure that we collect the data in the real world setting, then we would be able to select out the patients who are benefiting most and try and recognize when we should introduce CAR T-cell therapy at which stage of our patient's management.
How about your opinion about the CAR-T treatment in the first-line treatment of lymphoma? Thank you.
For first-line treatment for CAR-T, I think that we would have to make sure that the safety data were better than we have at the moment for the currently approved CAR-T, because we do send a significant number of patients to ITU with CRS and neurotoxicity. So, 60 to 70% of patients have a durable remission with standard R-CHOP treated chemotherapy, and I think that really I can't see it personally as first-line treatment unless we are better able to select out those patients in the future who aren't going to respond to R-CHOP frontline treatment.
How about the opinion about the PD-1 antibody in the Hodgkin lymphoma? And for the relapsed and refractory Hodgkin lymphoma, anti-PD-1 therapy is a standard treatment for the Hodgkin lymphoma, but it's very effective because of the relapsed and refractory Hodgkin lymphoma has a high percentage of PD-L1 and PD-L2 expression, and I think maybe for the untreated Hodgkin lymphoma, maybe the PD-1, PD-L1 and PD-L2 expression is less expressed in the Hodgkin lymphoma compared with the relapsed and refractory patient. And I don't know if maybe for the PD-1 PD-1 antibody treatment, maybe is more effective in the refractory and relapsed Hodgkin lymphoma than in the untreated Hodgkin lymphoma. How about your opinion?
I think where we fit PD-1 treatment in Hodgkin, again, is difficult because we get excellent response frontline with our standard frontline treatments for many of our Hodgkin patients. So again, if we could pick out those patients up front who weren't going to respond to ABVD or escalated BIACOP, then we might want to introduce a checkpoint inhibitor early on. However, at the moment, I'm not sure we would want to expose all of our patients up front to checkpoint inhibitors because it's not necessary for all of them, and if there are no specifically poor prognostic factors for these patients, we may want to save it for later on relapse. The other issue is choosing when we use checkpoint inhibitors is that we often don't see a complete metabolic remission with checkpoint inhibitors in Hodgkin lymphoma, and obviously that's something that we're all trying to obtain prior to using autologous stem cell transplant consolidation for our patients with Hodgkin. So I agree. I think that at the moment we're not sure the sequencing of a lot of our treatments that are available, when we should use brentuximab, when we should use checkpoint inhibitors. At the present in the UK, they're funded at certain lines of treatment. And we are currently undergoing clinical trials looking at the outcomes for patients using it either frontline or earlier on in relapse.
Okay, thank you very much. Thank you.
Thank you.
Congratulations for your Alexander study.